Our speaker this afternoon is Dennis VanEngelsdorp, probably well known to everybody in this room. My reason for bringing him here was this Varroa and his role in the VIP program. And I'm going to ask him, if he doesn't tell me, I'm going to ask him a lot of questions about that, because everybody in this room should be a part of that program. And if you're not today, I'm not going to let you leave until you sign up. All right? All right?
All right.
Dennis.
Great. Thank you. Good afternoon. I feel like I'm on Hajj. I've gone to the Mecca. This is my first time at this place, and of course it's famous in American history, so I feel really privileged to be here. I have to admit it's been a rocky 26 hours, because 26 hours ago, every Friday I meet with half of my students, so for half an hour each. I went through 4 students, and I finished up my talk, and I thought, well, I better look up to see exactly how long it is. And so it was an hour talk. I thought that was good, but I didn't know exactly. I better check it, and it had 3.5 hours. And I went, only a crazy person would ask you to talk for 3.5 hours. That has to be a mistake. So I went upstairs to pour my coffee, and I go, wait a second, Kim just invited me. I said, well, maybe, okay, Kim's asked, he's not crazy, but surely no one else would agree to 3.5 hours. And then I thought, wait a second, Randy Oliver is there, John Miller, Andy Card. I better call Kim and make sure this is a misprint. And he told me dutifully that it wasn't a misprint. So it actually worked out good, because I had to delve into some of the deeper bee biology or the Varroa biology than I have in a long time. So I'm excited to share some of those things with you in today's talk. So as Kim mentioned, I'm part of the Bee Informed Partnership, and we conduct the loss survey every year. We've done that now for 9 years. And 5 years ago, we switched that survey, so we started talking about not just winter loss, but also summer loss or annual loss. And what you can see, and it's really quite troubling, is that our winter loss, it was up at 30. We had just over 35%, and our winter loss, which is the yellow, has come down a little bit over the peaks we saw early on. But if you look at our summer loss, you can see that has now surpassed our winter loss. And so now we're in a situation that last year we lost more colonies between April and October than October and April. Now that's astonishing. I mean, that's the time that bees should thrive. There's lots of good food out there. It's a great time for bees. And so it's really troubling that we're seeing these high losses in the summer. This is the annual loss, of course, but you can guess what the summer loss is over there. We also asked beekeepers what they think is a reasonable loss. What would be reasonable to expect, what you should have expected to lose over the last year. And this is really surprising too, that on average, beekeepers say that they should lose about 15% of their colonies. That's what they think would be a reasonable rate of loss. Can you imagine a cow farmer saying, oh yeah, if I lost 15% of my cows over the course of the year, I'd be happy? And so this is troubling. We've come to accept these very high rates of loss in our industry, and I wonder whether we need to. I wonder whether we have to really think of even these, what we consider acceptable rates of loss, whether those really are acceptable. I mean, traditionally, before Varroa mites, before the 80s, the 1980s, people who lost 10% were too embarrassed to tell their neighbors about it, and now we're happy with 15% in some cases. So certainly there's been a big shift. And if as beekeepers, you know that this shift and this whole idea of colonies dying has really captured the public's imagination. And it's meant that we have a lot of people, not necessarily beekeepers, who have very clear ideas of what is causing these losses. Very strong, very clear ideas. And I'm reminded of this saying from a Baltimore critic when I hear this, that for every complex problem, there is an answer that is clear, that is simple, and that is wrong. And I think that we have to remember that. The drivers of colony losses are complex. There's several different factors interplaying and playing off of each other, and we have to remember that it's broader than just one issue. Although I'm sort of mandated in this talk to talk about Varroa, and I certainly think that Varroa is a major, major contributor to colony losses. And I want to bring up some of the caveats, some of the sort of the qualifiers before I give a talk. And the first one is that I recognize that when I say I think Varroa mites are the major cause of losses in the United States, a lot of people get ticked off at me. They get annoyed by this. You know, I'm a beekeeper for 30 years, I know what I'm doing, don't say that. And I have to remind you that I'm approaching this whole idea from the point of view of an epidemiologist. And an epidemiologist is not someone who studies individuals, but studies the population. And so when I'm saying that, I'm not talking to you, I'm talking to your neighbor, not you. I'm talking to your neighbor. And so it is, in fact, very, very true that there are people who have had clear losses because of very poor nutrition. It's been a bad year. It's very true that people have lost lots of colonies because of pesticides. And if you're that person, that's the only thing that concerns you. But from a population point of view, I'm hoping that over the next couple of hours that we talk and I convince you that Varroa is a bigger problem than I was expecting. I think that I'm surprised at the level of Varroa mite and how quickly we see Varroa mite populations increase at the population level. The other caveat is that a lot of the data that I'm presenting is correlative data. And correlation is not the same as causation. If you want to find the best predictor of colony losses based on correlation, it's actually the arrest rate, the number of people who are arrested for marijuana possession. And so it's inversely proportioned. Now, of course, when you see this statistic, you all know that there is nothing related between this. This is a fluke. This is not causation. There has to be a biological mechanism there. Now, sometimes things are covariate, so that they're correlated. So, for instance, there's a very strong correlation between shark attacks and ice cream sales. And that's a very good, strong correlation again. But of course, it's only because you swim in the summer, which is when you eat most of the ice cream, that that correlation exists. And so I say this because a lot of the data I'm about to show throughout this study is correlative data. Now, the preponderance of evidence, once you've seen correlations year after year, or over different populations, or in different situations, then you can become convinced. But just on their own, they are not data. You need some experimental data to justify it, or you need a lot of other correlative studies to help. For instance, smoking. We have no experimental data that smoking is bad for you. It's all correlative. And the reason for that is it would be unethical for me to cut this room in half and say, okay, you guys smoke 2 packs a day, and you guys don't, and let's see what happens. So sometimes things aren't set up to do good experiments with because they'd be unethical or too costly. So correlation has a value, but it's not the be-all and end-all. So I want to emphasize that as we work through. Now, I think as you've probably heard already, there are many different things affecting colony health. There are these interacting factors, and generally we focus on 3 big factors, maybe 4. There's the parasites and pathogens, and of course that's what I'm going to be talking about, is the Varroa mite. But we also have pesticides, both the pesticides farmers are applying and the pesticides beekeepers are applying to control Varroa mite. We also have poor nutrition because of monoculture, habitat, or just the lack of good forage around there. And then out here we also have this factor in management. And of course, I'm not going to touch on these. You've heard already stuff from Randy about nutrition. You'll hear a little bit more tomorrow about management and some of these other factors. But we're really just going to talk about this parasite in terms of Varroa mite. Now, one of the things we do when we ask the winter loss survey, and we do the summer loss survey, and we do that annual loss survey, we follow that up with a management survey, and I'll describe some of those results later on. But we also ask the beekeeper themselves, what do you think was the major reason your colonies died this last winter? And right away, and we're going to come back to this again and again, you'll notice that the response differs between subgroups of the beekeepers. Backyard beekeepers have very different responses than commercial beekeepers. Backyard beekeepers we're defining as people who have 50 or fewer hives. Commercial beekeepers, we think 500 or more hives. And so if we look at backyard beekeepers, they identify a poor winter pretty consistently as the major cause of loss, followed by starvation, followed by having weak colonies in the fall. So these are all manageable conditions. These are things that you can manage out of in many cases. However, if we look at commercial beekeepers, they identify queen failure, followed by Varroa mite, followed by pesticide as the leading 3 causes of losses. And so queens and Varroa mite interchange year by year in terms of whether they're ranked first or second by commercial beekeepers. But clearly, Varroa mites are identified by commercial beekeepers as being a problem. And these are guys who are actively managing. So they know what they're doing, they're actively managing, and they still recognize the problem with Varroa mite. And that's because this is sort of this silent monster in our midst. Talk about horror movies. You know, if you're a bee, this is what keeps you up at night. This is what keeps your children from sleeping, is this big, ferocious looking mite coming out from your mother's back, about to attack you. And it is actually a very amazingly well-adapted parasite for bees. I mean, it's really good at what it does, which is to hang on to bees, move into brood cells, and have more babies. And so we can look at its anatomical features and sort of respect why it's so good. The first thing you'll notice is that it has this really hard shell. It's a hard tick, in fact, and that hard shell keeps it from dehydrating. If we think of other mites, like honeybee trachea mite, it has to live in the trachea of the bees because it dries up really, really quickly. It can't stay out in the environment very long. However, these mites have this very thick shell, or this thick exoskeleton, which allows them to survive outside of the hive, or on the bottom board of the hive, or outside of the brood chamber for extended periods of time. The Varroa mite too, if you'll notice, has all these hairs here. These are sensory hairs. They can sense things. But they also have this line of really, really hard, nearly like nail-like hairs on the outside. And that helps deter, they think, it helps other bees from going in there and chewing on that mite. So it deters grooming. And it's those sharp sort of projectile spines that the mites have. There's also a wax coating on the mites, and that wax coating absorbs the smell of the colony. And so it's sort of like chemical camouflage. And so it smells like the colony. So you think about these colonies, they're really dark and they're really humid. And so now the bees would rely on a sense of smell to get rid of intruders. Well, this smells just like the colony, and it adapts well to the colony because that wax is able to absorb the smell of the colony. We also, if we look at the feet of the mite, what you can see is that they're a little bit different than bee feet in the fact that they still do have these claws, which are great for grabbing onto things. But you'll notice that they have also all these little hairs here. Honeybees have a pad there. It's like a suction cup pad that allows them to walk up and upside down. But these mites use these hairs. And in fact, these hairs are a lot like the hairs in a gecko's feet. And that's what's happening is it creates this really large surface area. And so, in fact, it's the attraction of molecules that keeps the mite able to walk on the vertical surface. It's actually causing this huge surface area, and so it's molecular bonding that occurs very weakly. But because the mite is so light, and because of that tremendous surface area, it's able to crawl on the upside down or up the comb and sideways. And so it's got really good at moving around, both on the mite and away from the mite. It also has this special little tube here, and this allows it to breathe. We'll talk later on about how mites reproduce, and one of the first things it does is when it enters a brood cell, it buries down into the brood food, into that pool of liquid food that the bees are feeding, And so this tube then allows it to breathe while it's submerged in that surface. And so it's adapted for that behavior as well. What was I going to say here? Oh yes, and so we're just again how strong that carapace is, that really strong outer shell, that exoskeleton. It's very good. It's also very thin. And this then allows it to slip between the plates on the abdomen of the bee and get right in there and feed between that soft tissue that connects the hard plates on the adult bees. So now some of you are probably a little anxious and you're thinking, how am I going to sit here for an hour or 2 hours listening to this guy? And what you really came for is, yeah, you're convinced that mites are a problem, and what you want is the silver bullet. Like, just get to the talking point and let's move on. And so I'm going to do that for you. And what I'm going to do is I'm going to give, you're going to have to turn the film off, and we have to all vow not to ever mention any of this again. But I'm going to talk now about how to make your own Varroa treatments so you can control mites and not worry about it. It's totally illegal, everything I'm about to say, so you can never repeat it. And of course, maybe a year or two ago, the truth is that if you were a commercial beekeeper and you were asking me what you should treat, I would tell you to call certain commercial beekeepers. And that's because there were lots of different products that were a lot cheaper to make yourself and apply to the hive. Those days are over now, though, because the only two products that you can use and mix your own that's available don't work anymore. That's coumaphos and fluvalinate. They don't work anymore. Amitraz you can't buy in a way that you can make your own product anymore that doesn't have high levels of xylene in it, or you have to smuggle it into the country. And so there is no secret backyard recipe. There just isn't. And so it's not even possible to do this if you want. So then you're thinking, well, tell me about what's about to happen. What's going to happen next year to save our day? And while there is certainly research out there that may be suggestive and may be helpful, the fact is that a lot of those products are years away. We're stuck with what we have right now, and what we have to do is figure out how to use it more effectively and more efficiently and in a way that's going to sustain until some of these new products come out. And so here I'm going to come up with this last little quote, which I think is really useful, and is that there is no silver bullet, and frankly, you probably don't need one. It is far more important to be able to find the right kind of gun, be able to load the gun, and perhaps most importantly, be able to figure out where the werewolf is. And I think that's a really good quote, because really what we are doing is we are hunting mites in our operation. And in order, how many of you guys are hunters or fishermen? And so if you are a hunter or a fisherman, you know that the first thing you need to do is figure out the behavior of the fish. I'll never forget, I had one of the first people that worked for me, Mike Andre. He was a trout fisherman, and he would go fly fishing. And he could talk for hours about how he would select his fly. Sometimes they had to be tied green, sometimes they had to be tied brown, depending on what grasshoppers were jumping into the river. And so he had to know exactly how that fish behaved, what they were eating at that time of year, to know how to catch those trout. And I think that for us, what we need to do is really have a very comprehensive and good understanding of the mite and mite biology in order for us to more effectively treat our mite problems. And so what I want to do for this talk is break it up into sort of 3 sections. That first section, we want to talk about the behavior of the mite. So like if we're thinking about werewolves in that analogy, we need to understand what makes a mite a mite. We need to understand what the behaviors of that mite are, where the vulnerabilities are, and then we can talk about how we shoot or get rid of that mite. So we're going to basically have 3 sections. We're going to have a break between each. One, we're going to talk about the life cycle of the Varroa mite and look at where it belongs sort of in the tree of life. And that might give us some insights about how it behaves and what it does. Then we really want to spend some time understanding the population dynamics of Varroa mites. Especially, I want to move away from just thinking about Varroa, because we're probably, I think that some would argue that mites aren't the things killing your colonies. It's the viruses they transmit that are killing your colonies. And so we have to talk about this in context of the Varroa mite virus complex, that these are these partner organisms causing the damage. And finally, we're going to conclude then with some treatment options and strategies. It's really slow passing. So this is the tree of life in the eukaryotes, or the more advanced, so it doesn't include bacteria, and it doesn't include some of the archaebacteria, which live in thermal vents. Those are sort of branched off into their own kingdom. But if we look at the animal kingdom, what we can see is here's the protists. That, of course, includes Nosema is a protist, a type of protist that became the fungi. And so we can see the different things, and here we can see the vertebrates breaking off. And there's this branch over here, right after mollusks, where what you can see is we see the crabs and the crustaceans, and then over here we see the arthropods. And so this whole line here is the arthropods. I can't say enough about arthropods. What percentage of life on Earth is an arthropod in terms of species? Any guesses? 80% of life on Earth is an arthropod. Compared to humans, how much do you think, for every human, how many arthropods do you think there are? There are over 100 million ants for every single person on Earth. Ants alone, which is one group in this group, ants alone weigh more than all the mammals, so that includes people and cattle and elephants and all the wildlife and all the trees. It weighs more than all of that, then ants weigh more than all of those things collectively. And so we're talking about, in many ways, one of the most successful organisms on Earth. These groups, these arthropods, defined by these hard exoskeletons where the skeleton's outside, are the most successful organisms on Earth. And of course, they include both bees and mites. And if we look at the extant, which means existing or living arthropods on Earth today, what you can see is that it includes the spiders, or the chelicerates, and that's where the mites are. They're over here. Recently, they thought that the spiders and insects were really closely related. They've reanalyzed that now, and insects actually, and and spiders are very distantly related within the arthropod group. So they're not as closely associated. These, of course, are the centipedes and millipedes, our crayfish and our crabs, and then the insects over here. And so they're part of the chelicerate. The mites are chelicerates. And so what can we learn about mites when we just look at that broad category of spiders? Well, first of all, this class has a whole bunch of different organisms. We have scorpions, we have true spiders, we have sort of these weird spider scorpion looking like things, dandy long legs, and over here we have the ticks. And these are a lot of them. There's over 100,000 of them. And these mites are extremely diverse, as we'll talk about later on. And so they're defined by the fact that they actually have 6 pair of appendages. So they have 12 appendages. 4 of those are legs, of course. We all know that. Spiders have 8 legs. And then right here in that blue, well, I guess that blue is the pedipalp. And so that we'll come and look at that more closely. And then we have inside there the mouth parts. We call that the chelicerae, after which they're named. They have simple eyes. They don't have compound eyes, which means they don't see very well. They might see some light, but they certainly don't see images. And they mostly feed on liquid external digestion. So they're digesting their food outside of their body and then sucking the nutrients in. And so if we look at the chelicerae, these are really quite admirable. You can see this one. They're tipped with fangs, so they're really good for horror movies as well. And scorpions have evolved, their chelicerae have evolved, so they're little pincers. And of course, what they're doing is they're going to put their fangs in and spit into the organism they've grabbed, liquefy it, and then suck up the juices. And that's how they're going to get their food. And then surrounding those chelicerae, they have these pedipalps. And so these pedipalps look like these sort of stunted legs. In spiders, the males actually have well-adapted pedipalps like this, and that's because they actually ejaculate into the pedipalp and then just swap that into the female like this. And so they've modified that as their mating organ. And it actually springs out, so sometimes you can see a mated spider because you see this pedipalp hanging out of her still. But mostly, for most organisms, these pedipalps have these hairs, and it's those hairs that are used to smell, it's those hairs that are used to sense the environment and walk around the environment. So now I want to show this video of of a mite, a tick, a deer tick in this process feeding, because I think it sort of tells us a little bit about how Varroa mites might do it. So you can see the two pedipalps on the side there, they're getting pushed aside, and that chelicerae moving in, and now it's sucking up. But watch, what it's doing is it's sucking in and realizing it's sort of like a straw. You know, your straw gets plugged, what do? You blow out, right? So the tick does exactly the same thing. It starts blowing out or spitting out, and it's actually releasing enzymes into the flesh of this mammal, because this is a deer tick. And then eventually it does that back and forth until it liquefies and this blood becomes easily accessible, and then it can suck up that blood. And so this is a tick, which mites are, Varroa mites are, and so mites probably do that same sort of behavior. But what they're doing is their pedipalps, they're finding the right place, and they're using their chelicerae to cut through that exoskeleton of the bees in order to suck up the liquid. And so if we look at a close-up of a Varroa mite, what you can see is these little pedipalps here, and then the chelicerae here surrounding the mouth parts that are going to use to suck up the juices of the bee. So we have all these mites and ticks. There's over 50,000 species of these. There are at least 1 million. They estimate there are at least 1 million, and there are just an amazing diversity of these mites. Most of these mites are soil mites. There are millions and millions per acre. In fact, there's some evidence to suggest that trachea mites started out as soil mites that then transferred over onto bees and started living in the trachea. And so sometimes we actually do get, when people are worried about having sort of the new mite that's killing all the bees, we'll get samples, and undoubtedly those are soil mites that just the bees accidentally brought to the colony. The hard tick, which is, you're probably familiar with some of these hard ticks, the hard tick which the Varroa mites are part of, this is, of course, this is how they look for their host. They're sitting out there with their front legs out there, and mites do this too. If you watch Varroa mites looking for a host, they have their legs sort of waving in the air, waiting for the host to come by. They feed on the host. They have these gonanophores, or nanophore, which is the sort of the mouth part that gets drilled down there between the chelicerae. And then when it's fully engorged, the female will drop to the ground and lay all sorts of eggs. And in the case of deer ticks, what you can see is those eggs hatch and they become larvae, and they'll get on mammals or birds or people, and then fall off again, become nymphs, get onto another host, fall off, and then get onto deer, other hosts, and fall off as engorged and lay eggs. Now when you see this pattern, where you see that same mite goes to different hosts, between hosts, that means that this is a great vector of disease. Because if any of those animals in that whole cycle had a disease, then it could suck that disease up into the blood and then pass it on in the next feeding. And so when you see this sort of behavior where you're seeing a mite feeding on more than one host, you expect it to be associated by vectoring some disease in those populations. And that's certainly true for Varroa, as we'll see later on. Mites feed on multiple different generations of bees, and as a result, it's a great vector of bee diseases, specifically viruses. Of course, in this case, it's Lyme disease. I think beekeepers really have to be careful. This year was a really bad year for deer tick where we are. I didn't go out once, but come back and find some deer tick on me. This, of course, you always want to be on the lookout for these markings. Has anyone ever had Lyme disease? I guess you don't want to confess that, maybe. It's an awful thing to get. So if you ever see this sign, you really want to go and get some antibiotics, because this is a terrible thing to live with. Now, in terms of bees, there are at least 86 different mites that live with bees. Very few of them are, in fact, parasites. Most of those are scavenger mites, and so they live just in the bottom board and they're just going around and they're eating pollen. Sometimes you'll get heavy levels of these pollen-feeding mites in your pollen frames. I've seen this in Florida where the pollen comes out really powdery. Have you ever stored frames of pollen and it gets really powdery and misty? Well, that's probably because you have a heavy pollen mite infestation in there. That also affects some of the native bees or the stingless bees, not stingless bees, sorry, in this case the carpenter bees, where what you can see is, or mason bees, you can see the pollen all shredded up into dust, and that's because there are these pollen mites in there. And so you have pollen mites, and then you have mites that eat the pollen mites, and so you have these predatory mites that eat the pollen mites, and then you just have hitchhiker mites that are just basically using the bees as a bus to move from flower to flower and then eat the pollen on those different pollens. And so that's most of them. But then, of course, we have our parasitic mites, which feed directly on the bees and the bee brood. One of those, of course, is the trachea mite. This came in the early 80s. It really did change our industry, this little mite. And what they do is they live in the tracheal trunks of the honeybee and make it difficult to breathe. It makes the bees very difficult to overwinter. But something happened, either because Varroa mite came and our control products for Varroa mite also helped control the trachea mite, or, and I think this is more likely, that the genetic changes needed by bees in order to keep these mites low happened really quickly, and it was a simple genetic switch. We very rarely find trachea mites at all anymore, and they're not an issue. We don't ever find them at high enough levels. We don't even monitor for them anymore. It was too much work to monitor for how few we find them. The mite we are really worried about finding is the tropolalaps mite. This is an Asian mite. It's a small mite compared to the Varroa mite. It runs around on the comb very quickly. It's oblong. It's throughout Asia. And I will say that on European honeybee colonies that are kept in Asia, they don't talk about Varroa mite. They only talk about tropolalaps. They're going in and having to treat every 2 weeks to keep this mite in check. And so this is one of our biggest concerns, is getting this mite into the country. This is be another huge game changer.